Okay, hello everybody, and welcome to the EXPBIO webinar on regional seed mixes. My name is Durka. I'm the head of the working group Biodiversity and Evolution of Plants at the University of Oldenburg, and I'm part of the EXPBIO consortium, which is a program financed by the European Union, the North Sea Interreg project, and we are interested in how to restore our landscape with seeds, with plants, and especially native plants that are regionally specific. And for that purpose, we are especially interested in the use of regional seed mixes. And today in this webinar, we welcome Dr. Walter Durka from the UFZ in Halle Leipzig, who is probably the one that can best tell us how the regions we define by landscapes relate to the genetic diversity in plants. To give you some feedback, background on Walter, he is studied in Bayreuth and worked on vegetation and the influence of nitrate, and then 30 years ago went to the I'm not sure what's the English exact name of your institute, Walter. Environmental Research Center. Oh.
It's Center for Environmental Research. It's a Helmholtz Center.
Right. And in his career at the UFZ, he has delved more and more into the genetic analysis of plants, population genetics, phylogenetics, and yeah, especially in the last years, concentrated on the population genetics of germ plants and beyond. And we are very much looking forward to hear what you have to tell about the genetic diversity in our landscape and what this means for the use in regional seed mixes. Thank you for joining.
Thank you very much, Dirk. So I just try to share my screen. Let's see whether this works. Which one do you see? Do you see the proper first slide of my presentation?
No, this is just the PowerPoint view. Okay. Not the presentation mode yet.
Not the presentation mode. Sorry, then I have to
share the screen the last time, Walter.
Okay, so I try to do it again. Oh, maybe it's this one. Yeah, okay, this seems to be the right one. Do you see the first slide of my presentation now?
Perfect.
Okay. Yes, so welcome. I want to inform you about our project RegioDiff, which was a nationwide assessment of genetic variation of grassland plants in the context of an existing seed transfer zone system. And I give this presentation together with Stefan Michalski and Johannes Höfner, the PhD student of the project. So what motivates us? Of course, we want to conserve biological diversity, and as population geneticists, in particular, the diversity within species, the genetic diversity. At the same time, we are aware that biodiversity is endangered and that restoration is a big task for all of us. However, if you look into the German nature conservation law and how this is all implemented, there is not much to be found except of one paragraph, paragraph 40, which says that the spreading of plants into the wild outside their areas of occurrence is not permitted. Genetics in particular is not mentioned in the nature conservation law very much, but this is a tool, so to say, to define regions of occurrence that make up genetic sense. However, these areas of occurrence are not also defined in the nature conservation law, but in fact, in another ordinance, where seed zones have been defined. So let's look into this seed zone system that is actually defined. It was defined by a project at the University of Hanover in 2010 by Prasse et colleagues. And here you see the map. These are 22 seed zones within Germany. They are based on ecoregions, which have been defined in the last century already. This is the system of these ecoregions, Naturräume in German, and more or less the seed zones represent these ecoregions. They are based on climate, soils, geology, landscape structure, also a bit on biogeography. The system of these 22 seed zones is legally binding in a sense that if you want to restore in the free landscape, you have to use seeds that have been produced using this scheme, which means within a seed zone, seeds have to be collected in a number of sites. They are mixed. They are propagated on agricultural fields, and then again are used in seed mixes to restore a particular site. As you likely know, there are seed producers in Germany, at least 2 large ones. They have their certification systems, and there is a growing market. So the system actually works in Germany quite well. So what's the problem? So first, already more than 10 years ago, we were interested in the question, okay, there is the system. Is it actually doing what it should? Is there local adaptation? And we did a common garden experiment with material that we bought from those seed producers. So we had 7 species and 4 common gardens, and we looked into biomass and flowering and seed production and things like that. And we found out, without going into the details, we found out that there are phenotypic differences between material from different seed zones, and that these differences increase with geographic distance and with climatic distance, which together told us that regional adaptation actually is present. However, we had only 7 species and only 8 of the regions were included at that time. So the seed zones in general can be questioned because, as I said, they were defined only based on ecoregions and without any knowledge of adaptation or genetics. And the seed zones apply uniformly to all species. So it's not species specific, but it's a general system. And the seed producers have criticized the system because at least some of the regions are rather small and may make problems economically. So therefore, our project was initiated by the Federal Agency of Nature Conservation with a principal aim to first have an empirical knowledge base on the genetic variation of species across Germany. In particular, for the seed producers, they wanted to know whether one could build species groups and have different systems, for example, for grasses in contrast to herbs. And in general, we were asked to suggest changes to the system if appropriate. So what did we do? This is the whole scheme I will lead you through. First, we had to define a list of species we were interested in. So total grassland flora in Germany is about 1,400 species. The seed producers produce around 400 of those. And in the typical seed mixtures, around 200 are actually make up the largest part of the species list. We wanted to be representative, which means some grasses, some herbs. We wanted to have many of the average species, but also some from the wet and from the poor habitats. In the end, we came up with 28 taxa, and this is the list. And you see already that some of those have different ploidy levels. That means we did not exclude problematic species. In particular, we included things like species like Achillea millefolium aggregate or Leucanthemum vulgare aggregate, but also the other species that are listed here, which have 2 different ploidy levels included. These are the species again with a number of sites sampled. So we were looking for people that could us help sampling. So we had 160 volunteers. the 22 regions were dissected in a total of 72 sub-regions, and each collector or collector team was responsible to collect one of those sub-regions. The first aim was to have 5 samples per sub-region in as natural as possible grassland. Overall, we received something like 19,000 samples from 2,500 sites. Then we selected in the first step only 1 sample per site and species, which made about 12,000 samples. And we did SNP genotyping for all those species. SNP genotyping means we have single nucleotide polymorphism data, so it is sequence data, and those SNPs, we don't know about the specific gene functions of those because none of our species has actually been sequenced, or at least we didn't use the sequence data. The basic assumption is that if populations are genetically differentiated, this is maintained by a lack of gene flow, then adaptation can take place without being swamped by gene flow. This is just an overview about the lab method. If you are interested, we can go into that. What is important is that we have about 5,000 SNPs per species, from 1,500 to actually 12,000 in some of the species. So this also differs, but we have thousands of genetic loci on which our analyses are built. What you will see in the following is mostly principal component analysis. So these upper graphs which show the genetic distance of the individual plants. And these in the center shown genetic structure analysis. This is a so-called admixture analysis, which defines genetic clusters of each individual, which can be either one or more genetic clusters mixed within one individual, which data then can be put on a map by kriging and spatial interpolation to actually have a genetic map of genetic identity. We were also interested in isolation by distance, asking whether genetic differentiation is correlated to geographic distance. So let's jump into the results. I start with a very common grass species, Arenatorum melasius. And what you see here is a principal component analysis. Each dot is a plant. The color code represents the seed zone. In this particular species, we see a big mess. There is hardly any structure. Only the seed zones 18 and 19 are a bit off of the rest, which means there is hardly any genetic structure in that species. If we do the admixture analysis, we can identify a little bit that in the southeast there is one cluster more predominant. This is what we see also with this seed zones number 18 and 19. So the Alps and the Bavarian forest are somehow separated from the rest, but not very strongly. Still, however, if we look for isolation by distance, this is the correlation between FST, the genetic distance, against geographic distance. We see a flat but significant correlation. So the further you go, the more different genetically you become. So in this species, we have very low structure. If we look into this bar plot at the bottom, we see that many of the zones are actually mixed and there is not much structure. So you would say, okay, that's a grass, it's a very common grass. We know that it's not very old in our landscapes and therefore this is understandable. So let's have a look into another grass, Acrostis capillaris, also very common. And here we actually do see much more structure. We see the green and the blue dots. This is northern Germany in contrast to the other colors, which is more the southern part. So there is clear differentiation geographically. If we do that mixture analysis, we clearly also do see this. And surprisingly also, there is more structure in the north than in the south of Germany, which is a bit surprising as northern Germany is more flat and more equal compared to the more structured south of the country. Again, looking for isolation by distance, we see a very nice correlation, which is also much steeper than for the other grass. So we see sorry, more structure and differentiation in the north of the country. Let's move to Acrimonia, a herb, which again shows very clear structure. In this case, the northeast of Germany is very off compared to the rest. So there is clear geographic structure. And we look into the admixture analysis, we also see quite nicely the different regions are separated. So these clusters that we can identify with admixture are geographically defined. If we look into the distribution map of exactly that species, we can see that distribution gaps, like in the Franconian and Bavarian forest, lead to separation and differentiation of the genetic pools. So there is no gene flow across these mountain ranges, and therefore regions north or south of mountain ranges are genetically differentiated. Again, there is isolation by distance, in this case not as strict as in the other examples, likely due to those mountain ranges which hinder direct gene flow. So we have, again, a grouping, in this case, of 6 groups. We see that distribution gaps are genetic barriers, and again, we have isolation by distance and environment. Let's go into another example. In this case, I show Ranunculus acris, very common plant. And here we first have a look not on the total DNA pattern, but on the pattern that is shown by chloroplast DNA. And chloroplast DNA is inherited only maternally, meaning it is inherited by seed. It's not inherited by pollen flow. And traditionally, any patterns seen in chloroplast DNA can be interpreted as of being historically and related to colonization patterns. And so if we look into the chloroplast pattern, we see, okay, there seem to be 2 chloroplast groups, the blue and the orange one. And if we plot this on a map, we see that one of the groups is only present in the west, and the other is only present in the more eastern half of Germany. And this very nicely matches with what we know about postglacial colonization of Central Europe, which can or which could have taken place from the west, from the south, or from the east, or even from the north. In this case, it could be the case that 2 colonization routes exactly meet within Germany from east and west. Looking again for the same species on the pattern of the whole data set, we again see a nicely separated group. And if we look again for the separation at k equals 2, if we only look for 2 clusters, it's again the same pattern that we have just seen, east versus west of Germany. If we go up and look for more groups or allow for more groups, we can see much more differentiated groups. For example, in the very north of Germany, this pink group, which is clearly visible. So in this case, we have again isolation by distance, we have clear groupings, but we have additional influences which we can interpret as a signal of the phylogeography of the species. Let's come to another very common grassland species, Leucanthemum, in this case Leucanthemum irkutzianum, the tetraploid of that species pair. And if you look into the PCA graph, it looks a bit different than previously because there is a big group of points, of data points, and in the same seed zones we have these upper examples, these upper points of samples which are somehow different. If we again look into the chloroplast, we can understand this pattern. We see that there are 2 chloroplast groups in that species, and that this second group in the upper part of the PCA graph is exactly produced by one of those 2 chloroplast groups. So we know Leucanthemum irkutzianum is an allopolyploid, it's a tetraploid, it's allopolyploid, and obviously there are 2 different phylogenetic origins of those 2 groups which make the species and which complicates the overall genetic pattern across Germany. if we still try to do the admixture analysis, we can see some pattern which is not as clear as before, but also here we can differentiate 3 or 4 groups which have their respective ranges within Germany. Again, there is isolation by distance still to be found, although it's such a complicated case in here. And as I said, not only do we have this isolation by distance patterns, but we also see phylogenetic patterns, and in this case, the origin of the species by hybridization, obviously. Let's go even more complicated. If we go into Achillea millefolium aggregate, we know that this is a species complex of different ploidy levels ranging from diploid, tetraploid, hexaploid to octoploid species. And if we focus only, and these are nicely separated in this PCA graph by these 3 groups. If we now focus only on those that are common on mesic grassland, so we exclude Cetacea and Pannonica, we still have 2 very strongly differentiated groups of samples. And if we go into the ploidy level analysis, we see that one group is tetraploid, these are the blue dots, and the other group is hexaploid, these are the yellow dots. So we have Achillea millefolium sensu stricto, the hexaploid are clearly separated genetically from the tetraploids. This is this Achillea culina and Achillea pratensis. If we ask, and these 2 groups, millefolium and Culina pratensis, have not been collected on purpose, but people just went out collecting Achillea. So most of, or many of these collectors were really botanists. So it is obvious that they can be mixed up very easily, and they are morphologically not easily, if at all, distinguishable. But genetically, they are clearly 2 different things. And as we know, they have different ploidy levels. If we look into the distribution, we see that both the tetraploids and the hexaploids occur actually overall Germany, but there are some centers of occurrence for the tetraploids in southern Germany, but also in central Germany, and some regions like the northwest of Germany, which is only made up by the millefolium sensu stricto. We also see that in the mountain ranges like Bavarian forest or Black Forest, obviously only the hexaploids do occur or are really dominant. So concerning seed mixes and restoration, this clearly says that we need to reconsider what is actually to be sown, and maybe as a first step, also the seed producers have to check what actually they do have in their production lines. If we want to go a little bit more into detail with these 2 tetraploid species, we can do that later. The last example I want to present is Campanula rotundifolia, where we also know that the species occurs in 2 ploidy levels, the diploids and the tetraploids. And if we look into the PCA graph, we clearly see that these are strongly differentiated without any sign of gene flow between the 2, which says functionally and biologically, these are 2 different biological species, and therefore they should be kept separate. If we look into the distribution, in this case, we very nicely see that they are parapatric. So the diploids have their range in the east of Germany, and the tetraploids are distributed to the southwest and north of that diploid range. This also very nicely fits to the overall distribution of the ploidy levels in that species. So in East Central Europe, there is the center of the diploids. To the west and north, there are the tetraploids. And if we go even further to the northwest in Ireland and Scotland, then the hexaploids take over. So all this goes without any morphological differentiation. So this is clearly cryptic variation. And if you look into the distribution within Germany, this also says that we need to take this into account in restoration and in seed production. Another point why we should take this into account during seed production is a thing called triploid block. So if you look into the graph to the right, so a diploid population starting from F0 to F1 to F2, this means if seed production is pertained over generations, then a purely diploid population just will produce diploids. And to the right, a purely tetraploid population will only produce tetraploid offspring. In a mixed population where tetraploids and diploids occur, there will be hybridization and triploids will be built. Usually triploids are non-viable, or if they are viable, they are usually sterile, which means the seed production of such a field or of such a population, such a restored population, is much reduced compared to a pure population. And therefore, for the seed production on the agricultural field, but also for the restoration side, we should avoid to have mixed ploidy populations. And this is not a rare problem. There are many species actually which have similar problems of having multiple ploidy levels. And therefore we developed a recommendation how these mixed ploid species should actually be managed. So first, only native cytotypes should be, of course, allowed in regional seed. And as a first rule, only the dominant cytotypes should be propagated and released within a seed zone. And using our data across whole Germany, we can now define for each of the seed zones which, or if ever, which cytotype is the dominant one. If, however, we have a parapatric distribution, this can be then managed in producing sub-regional seed for only parts of such a seed zone, and further rules if we want to do that. So this is now the overview about all the more than 30 species that we could identify. You see is that each species is different. So we have a species-specific pattern of those genetic groups. Also, the number of groups that we can differentiate differs between 2 and 8. However, there are some common patterns. As I already said, for example, in the very north of Germany, in Schleswig-Holstein, it is always a single genetic group, although there is 2 seed zones that actually meet in Schleswig-Holstein. Or to the west of Germany, in many or in a number of species, we have a new group emerging in the west of Germany in a number of the seed zones there. Also, in the south of Germany, there is a separation into east and west rather than south and northward. Overall, whatever patterns we see, they are not concordant with the current seed zones. However, there are also very common patterns. As you have seen before, in each species, we have looked for isolation by distance, and actually in almost all of the species, we find a significant relationship between genetic distance and geographic distance, which means in all species, there is a more or less continuous change of allelic composition across space. And thus, we can also deduce for the practical use of those. Although isolation by distance is a continuous phenomenon, we cannot define a clear distance rule, but in general, we can say that the borders that are exactly mapped on a map, they must not be taken too serious on a project level. So if a project does a restoration project in the vicinity of a border or crossing a border, then using seed from the other seed zone across the border should be allowed. We are, of course, interested in some generality that we learn not only species-specific things, and therefore we ask which of the drivers that we know actually affect the genetic diversity and genetic differentiation. So First of all, the mating system. Second, we were asked, can grasses be distinguished from herbs, or the poloidal level? These are the obvious potential drivers. In the figure you see here is I ordered all species according to their genetic differentiation among seed zones from top to down. So as before, Arenaterum is the species with the lowest differentiation. And I marked the grasses in green. And what you do see is, of course, there are grasses which are hardly differentiated, but similarly, there are herbs which are only very weakly differentiated. And in contrast, there is even a grass that is among the most differentiated species that we find. So it's not as easy as to say grasses are not differentiated. This is not the case. So doing that more clearly in a statistical model, we looked whether differentiation among zones is affected by the mating systems, and clearly the self-compatible species are more genetically differentiated in space than are the self-incompatible ones, which have higher rates of gene flow. The grasses and the herbs are not statistically significantly different in their spatial genetic pattern. And the diploids are more strongly differentiated than the polyploids. If we look for the diversity within seed zones, there is not much pattern except of that the mating system again says that the self-compatibles are not as diverse as the self-incompatible species. With this, we come to the last and biggest question: can we define alternative seed zones based on what we have found across the more than 30 species? Again, we did quite some effort, and the steps were first put all the data into one big data set across all species, across all Germany, do some algorithmic clustering, allow also for expert proposals, rate each particular map that will result, define a benchmark in which we can actually rate those results, and in the end, hopefully discuss with stakeholders and come up with a meaningful solution. So what we did is we put all the PCA analyses of all species into one big matrix, which you see here, this blue-red matrix, which defines for each species and each subregion the PCA score, so a particular genetic identity. And this can be then subjected to a clustering algorithm, resulting in what you see here. This is the automatic clustering of the genetic identities of more than 30 grassland species across Germany. So for each assumed number of clusters, we find a particular map. And the question is now, how do these particular maps actually represent the genetic variation behind it? So we developed what we called explained genetic variance. It's kind of an AMOVA analysis averaged across all species. And this parameter says how much genetic variance is explained by a particular map, considering all species. We also can use the current map of the 22 seed zones to actually analyze how much variance is explained and use this as a benchmark. And the current map explains about 70%, so 0.7 of the genetic variation across all species. If we look into these different automatically generated maps, we see that this parameter is steadily increasing up to, say, 10 to 15, and then is only increasing very slowly towards higher values, which means our current map of 22 seed zones explains 70%. Using an algorithmic solution with also 22 regions, we could explain even more. We could explain something like 0.75, so 75% of the variation. And using an optimized map, we can explain the 70% using only 16 regions. Therefore, we look into the 16 region solutions and try to identify general patterns that we see into those. So first we have to recognize that there is no single best map. There are thousands of different maps that we could produce. All are a bit different, but the general outcomes of those are that the seed zones that were clustered, they are spatially coherent without using any geographic information. So only based on the genetic patterns, we get spatially coherent zones. The different zones are more or less similar in size. They are more or less roundish. Previously longish regions are separated into more roundish entities. What we also see is that the mountains and the forelands are mostly kept in the same cluster. They are not differentiated, which mirrors what we have seen before, that the forelands of mountain ranges are actually in the same cluster in general than the mountain ranges. Again, we see some particular groupings, like in the very north, Schleswig-Holstein is always the same group, or in the west of Germany, we see particular regions that pop up there as genetic groups. Still, overall, we see that whatever we see here as potential new zones, they do not match the current zones. We then went on together with seed producers, considering that they wanted to have less zones, ideally aggregating existing zones to come up with an easier system, a more simple system of zones. And the map that you see here is one of such a compromise to, on the one hand, keep zones as far as possible existing without changing them. On the other hand, aggregating existing zones into new and bigger zones. But still, we wanted, of course, to increase the genetic representativeness of the whole system, and therefore some real changes had to be proposed. Finally, there was a stakeholder workshop over 3 days in which we discussed potential changes to the system and what this would mean for the seed producers. And in the end, we came up with a decision that whatever change is made to the system would harm the availability of seeds, because seed producers would have to actually change the running system, which was felt to be hard. On the other hand, we have seen that the system as it is already encompasses 70% of the available genetic variation, and it's difficult to actually get more represented in the system. Therefore, our proposal is not actually to change the map. With which I come to the take-home messages. We have seen that in each species we get genetic structures between 2 and 8 groups that can be defined in a species-specific way. That mixed-ploid species need some special attention. In general, we see isolation by distance as a common pattern, which then asks for spatial flexibility in applying those zones. We have seen that the current system actually represents 70% of the genetic variation, and therefore no change of the seed zone delineation is proposed. However, what we can do, I didn't go into that in detail, we can of course, using the genetic distances between regions, we can define exchange rules of neighboring regions which are genetically very close, so that management is easier. I didn't talk about the climate change thing. We can do later if you are interested. I just wanted to point out that a recent development is that in cooperation with the people from Luxembourg, we are actually analyzing plant material from Belgium and from northeastern France, asking whether the genetic groups that we have actually seen in Western Germany, how far they do extend to the west into those countries, in particular for Luxembourg, because Luxembourg is a very small country and they of course want to know from how far they potentially could get their seeds if they do restoration projects in their country. And with this, I am at the end of my talk. I want to thank my lab crew, the people who have done the cytometry on the polyploid species, all the collectors without whom this would not have been possible, our funding agency, and you for your attention. Thank you.
Thank you, Walter. That was really an impressive work. Lots of data. And yeah, we somehow wished that data would have been there before the establishment of all these seed zones.
Yes.
But it also demonstrates how species specific this is, and this is just for grasslands. So if you are interested in others, forests or water systems, then the system may look again completely different. Yeah. Are there any questions from the audience? Jenny asked, what did you want to say about climate change? Yes.
So I just again share my screen maybe. Is it this one? Yes, of course, this is a system, a purely descriptive study of what genetic patterns exist at the moment. And of course, the question is always, climate is changing and what to do about climate change and these existing predefined structures based on ecoregions defined 100 years ago, more or less. We did one example calculation, again, with a different species, Gallium album here, but it's just one example, one potential example, in which we know, of course, there is the demographic history, these genetic groups that we can define across Germany, and the effect of geography, so isolation by distance. These are of course, two major drivers of the structure. However, we of course know that there is soil, that there is climate, which also could affect or could have affected the adaptation of that species. And if we do a variance partitioning analysis, we do see that, of course, pure geography explains a bit. The pure demographic history explains quite a bit of the genetic structure. The pure environment, however, also explains part of the structure, so is somehow correlated with the existing genetic patterns. And large part of the genetic pattern that we see is somehow confoundedly related to geography, demography, and environment. So there is more than just the geographic distance and the demography. And the question is, how can we use this correlation of environmental variability and genetic variability to somehow predict what will happen in the future? As a first step, we extracted those genetic markers, those loci that show a clear correlative pattern with some of the environmental variables we do have. So we have bioclim data and we have the soil data, and we get say 46 adaptive loci that have some special relationship with those environmental variables. And we can use the current climate, 1970 to 2000, to actually map the genetic variation in the eyes of the environmental variables. This is difficult to explain. So what we do see here in the left map is the correlation of particular environmental variables with the existing genetic patterns. And we see some high values in the Bavarian, in the Schwarzwald and in the Alps and in some of the other mountain ranges. If we used exactly this relationship between genotype distribution and environmental variables and map it on the predicted climate for 2080 to 2100, this map will be slightly different. It's difficult to see, but there is a difference. And if we build just if we subtract these two patterns from each other, we come up with this pinkish map to the right, which is the genomic offset. So you could also call it risk of non-adaptedness. So if we use the correlation between genotypes and environment now and extrapolate it to the future, we will see that some regions are not as well adapted as they could be. These are the more yellowish regions. The problem with such a calculation is, okay, what does it mean? Where is the threshold? Where is it serious? And using a method developed by Susanne Lachmuth and colleagues, she defined a threshold above which we could consider the non-adaptedness as serious, and this value would be the set score of 1.0. All our values we found for that species are below this one threshold. So, and therefore, our conclusion in this analysis for that species is that we do not expect big threats for the species to become non-adapted in future, based on what we have seen with our genetic data. This is what I wanted to say about, now I've lost track. This is what I wanted to say about climate change, summarizing that in the one case study we did not see much endangerment.
Yeah, but it's just one case. And so why did you pick gallium album?
We picked it because it was the first species where we had enough data. There was no particular reason to take it. We could have taken any other species. We did in parallel habitat suitability modeling studies with gallium album, and also there we found that the predicted habitat suitability is also large in future if we only use the habitat model, so which somehow fits to the other analysis.
Right. Yeah. Further questions? One thing, yeah. Do you know if other countries in the world have done similar studies?
Yes, I know that currently Czech Republic does exactly what we did. They used a little bit different set of species, but a large overlap of the species. They didn't study the grasses because they feared that the grasses are too much affected by sowing and agriculture. But essentially, they do the same thing that we did. I'm not aware of any other country, except of course, there have been these studies in Norway using a number of species with AFLP 10 or 15 years ago, but I'm not aware of. There are, of course, some case studies from North America for certain regions and certain species, but I'm not aware of such an analysis which is really multi-species and across the whole area of a country. Of course, the study is restricted to the national borders of Germany, which does not make too much sense, actually. But for the applied question of the seed system in Germany, it is on the other hand, of course, the right scale.
Yeah, but I think it definitely makes also some sense for the neighboring countries to look at these maps. But you mentioned the sowing effect feared by the Czechs. How much of that effect did you see? How much of these offset individuals that do not belong to the geographical group that they should belong to did you find?
This is difficult because so we did not have reference, or we didn't have too many reference sample of, say, commercial accessions of those grass species. We did not include that. We couldn't also do that. On the other hand, we asked all our collectors to collect only in natural grassland, and therefore it is difficult to. Of course, we had these outliers where in a, say, purely red region, there is one or two blue individuals, which obviously do not fit. However, whether this is historical gene flow or current gene flow or sowing is very difficult to say, at least in the scale of, in the framework of our analysis with so many species and across all Germany, we could actually not go into detail into these questions. We just have to accept that there is some blurring pattern or some mixture which we cannot nail down to a particular reason why it is the case.
but it's not something specific to some horticulturally more interesting species, like, for example, like Cantamum, where I could imagine that this would be more of an issue like in others that you have investigated.
So we have one case, and actually this is Knautsia arvensis. Knautsia arvensis occurs in 2 ploidy levels, and we found that 96% of all samples were the tetraploids, which is what fits to the overall phylogeography, and the other 6% of diploids, they were scattered around Germany. We know, however, that the seed producers grew the tetraploids and sold, they sold the diploids. So we know exactly that some of what is restored, some restored sites, this has also been verified by other groups, some of the restored sites have been actually restored with a foreign cytotype, which is genetically very different. This cytotype flowers earlier, it flowers longer, it grows larger usually. So it's somehow a nicer plant compared to the wild tetraploid Knautsia arvensis, which may be a reason for it. And it occurs also, I must admit, it occurs also in very famous nature conservation sites along the Rhine and the Donau Valley. But this, I hypothesize, is also a very old restoration site where the dams of the large rivers have been sown in the last century. So this could be one reason, because at that time no seeds were available potentially, and only from southern or from the Alpine region some seeds were then used to green those dams. So this is a hypothesis, but at least in this species we clearly see that the dominant cytotype and a subdominant cytotype, which was actually introduced more or less by the seed producers.
Yeah, great. Sheila asks, is it also planned to perform a similar study with rare species?
Not from my side. So there is a parallel study on agricultural weeds, but not on rare species actually, because of course you could, so in principle the regional seed zone system is defined only for the common species. So you will find only common species because the purpose of the whole system is that you do not have to get an allowance to do some restoration project. If you use the regional seed mixtures, which are somehow certified by the system, you can just use them without any further allowance, which should ease restoration. And if you introduce rare species, this would have to be then, of course you can try to do it, but it has to be allowed by the Nature Conservation Administration then in each case. And therefore, in the system, there are no actually rare species involved.
I mean, one problem that may arise in such analysis is hybridization between different genotypes that approach each other. So how come is that? How often do you find these suture zones between regions?
So very often we find mixture zones where two different groups, which are not defined very, very clearly, mix across Germany. So actually it's more the exception that we have clearly defined and clearly separated groups. Very often it's kind of an admixture zone that crosses across large landscapes. And this is quite common. So I think that if you call it hybridization between those gene pools, this will be very common, and only in cases like I showed Acrimonia, so north and south of the Franconian forest, these are separated gene pools without any ongoing gene flow.
Any further questions? I think there was a lot of food for thought for us here, and many different aspects that can influence these patterns that you described, and something that has to be certainly considered in more detail in one or the other study. But in many projects of restoration, this will probably be too much to be considered. But yeah, there are no further questions, and we are approaching the end of our time. Then I would like to finally thank you, Walter, also for the talk and for the informative discussion. I think this added a very important aspect to our whole webinar on regional seed mixes.
Yes, and in response, of course, if you you are a very large consortium ranging, if I understood well, from Belgium up to Sweden. In general, I'm interested in extending the geographic range of our analysis. So, if there is any fieldwork going on in your project, we could maybe come together at a further point to see whether some extension of our geographic analysis is possible together with what you do.
Yeah, we have one project on Achilleas, and that I'm sure we will contact you and see what we can do about that. Right.
Thank you.
Thank you. And you're all invited, of course, to the next and last webinar of our series, which will be in 2 weeks. And this will be by Malte Conradi, who will introduce then the issue on evolution of these plants in these seed mixes and what that may have to do with our use of regional seed mixes. And I hope that I can see you all again at that presentation then in 2 weeks. So have a nice evening. See you again soon.
Thank you. Goodbye.